Good evening, everyone. Thank you very much for coming to the museum this evening. We're very, very pleased to have Chris McKay here this evening from the NASA Ames Research Center, who flew in this morning, so hopefully not too jet-lagged. It's very kind of Chris to come all this way to speak to us about the search for life on Mars and elsewhere in the solar system. Chris is extremely active in astrobiology. It's very difficult to summarize Chris's career in a very short time, so I won't try and do that. He did his PhD in astrogeophysics from the University of Boulder, Colorado, and then moved to NASA Ames, where he's been instrumental in many areas of astrobiology. His work is focused on the study of life in extreme environments. He's done research in Antarctica, in the Arctic, in the Atacama Desert, and other places as well. But he's also been involved in many other areas of planetary sciences, for example, understanding the environmental conditions on Titan and elucidating how the geochemical cycles work on that moon. He's proposed missions to Enceladus and Europa. He's been active in many planetary missions on science teams for the Huygens probe that landed on Titan. He's been on the science team for the Phoenix lander that landed in the Martian North Polar region and found the first evidence of perchlorates amongst many other discoveries, and is now on some of the science teams for instruments of the Curiosity rover. So Chris this evening is going to tell us about the search for life on Mars and give us an update on what Curiosity has been finding on Mars. So I'd like to thank Chris again for taking the trouble to come all the way to Edinburgh, and hand over to him right away. Thank you, Chris.
Okay, well, I'd like to start by thanking Charlie for inviting me to come. It's always a pleasure to come to Edinburgh. Actually, I'm part of the Scottish diaspora. My mother was born here in Edinburgh, and I think I have lots of cousins around. Hopefully none of them came to the talk tonight, but they're here, so it's a pleasure to come back. And it's also a pleasure to be here at the opening of the UK Centre for Astrobiology here in Edinburgh. I'm really excited when I heard the news and the activities at the mine, and Charlie coming out here should serve for a really good start. Charlie's done a lot of amazing work, and I think it'll be interesting to continue the collaborations between activities here and NASA. So as Charlie indicated in the introduction, I'm interested in life and life in other worlds in particular, but what I want to focus on tonight is missions to other worlds, and where we are on Curiosity, the Mars rover, and other near-term exciting missions. So let's go ahead. First, a little motivation. What are we searching for when we go on these missions to other worlds? We're not just searching for life. We're searching for what I call a second genesis of life. And what do I mean by that? Well, I mean life that's not on our tree of life. This is a diagram, the original one from Carl Woese, that shows the tree of life, the three domains, the bacteria, the archaea, and the eukaryotes. Animals, plants, they're all up in here. And it's an amazing and really quite beautiful result of biology that all organisms on Earth map onto this tree of life. Pay no attention to the viruses. We don't know how we map them, but they're at least in there somewhere. That's as I said, a beautiful result. It's not a necessary result. It could have been the case that that these three domains didn't link. They were distinct and separate, and we had three different types of life representing three different origins, but it's not the case. We have only one type of life on Earth. So what we're looking for, or what I'm interested in looking for, is something that's not on this tree of life, an alien. That's how I define an alien. An alien is an organism not on our tree of life, which is interesting. When I was a kid, an alien was defined geographically. An alien was someone from another planet. If you were from Mars, you were an alien. And if you were an alien, you were from another planet. But now we don't define it that way. It's defined biochemically. You can be from Mars and not be an alien if you map on our tree of life. It's defined biochemically. Similarly, you could be living right here on Earth, right in the backyard, and be an alien if you're not on this tree of life. When I tell this to people, they often ask me, well, doesn't NASA have a secret storage locker where we keep the aliens? And I say, of course we do, and I'm happy to show you pictures of the aliens, and here they are. I can tell you for sure that these creatures are not on our tree of life. So by my definition, they're aliens. So that's my job, in a sense, is search other worlds for alien life. So when I'm on a long plane flight, like coming over here, and the guy next to me says, well, what do you do? Chatty fellow. I say, well, I search other worlds for alien life. And then he leaves me alone the rest of the flight and gets some sleep. So it's a great job description. I like it. But why are we really interested in the search for alien life? Well, first and most importantly, we would then have for the first time another example of biochemistry. All life on Earth, as I just said, has the same genetics and biochemical machinery. That's nice, but it limits our ability to understand how general that is. If we could find something that's different, we'd have life 2.0. And imagine what kind of questions we could answer if we had something to compare life on Earth to. To my mind, that's the most important goal of astrobiology, to find another example of life, a second genesis. Search for a second genesis. But it also tells us that life is common in the universe. If it started right here in our solar system twice, independently, that would be strong evidence that life is common. And I added my little editorial comment, yay. It'd be great to know that life is common in the universe. I'm all for that. So this is what we're searching for. Where do we look? Where in our solar system do we look? I realized the original title that I gave Charlie, or that Charlie put in the announcement, was Search for Life in the Universe. Well, we're going to be a little more modest, and we'll just search for life in the solar system. So where in our solar system would we go? Well, there's Earth, our standard. Well, here are the worlds that I think are the good candidates. Mars, Europa, the moon of Jupiter, Enceladus, the moon of Saturn, and Titan, another moon of Saturn. First three are water worlds. They all have water or had water. The last one is interesting in that it has a liquid, but the liquid is not water. It's an organic liquid. And it's an interesting question, could life survive or grow in a liquid that's not water? Now, of all these worlds, I give Enceladus a star, a gold star, and only Enceladus gets a gold star, because Enceladus is the only world where we can say all the major requirements for life, for habitability, are met. It has liquid water, it has organics, it has a biologically available source of nitrogen, and it has an energy source. It's the only place in the solar system where we can check off what I call the big four requirements for habitability. And that's why it gets a gold star. Mars, we're still searching to see if it is or was habitable, and same with Europa. They might well be. We don't know that yet. So tonight, I'm going to talk about Mars and Enceladus. Mars because we have a big rover on it and we're getting exciting results, and Enceladus because it gets a gold star. Okay, so a little bit about why Mars. Why is it always on the top of the list? Most importantly, evidence for past liquid water, but also the presence of an atmosphere with carbon and nitrogen, and Mars is cold and dry, and so evidence of life can preserve there very well. So we might be able to find something there. This is the canonical picture of what Mars might have looked like 3.5 billion years ago. It's an artist's conception, obviously, of Mars with the amount of water that the geologists think is required to explain the fluvial features we see. So if you just take that water and put it on the current topography of Mars, 500 meters over the whole surface, let it relax in the current topography, this is what it gets. Now, this picture is a bit misleading because it looks so warm. It looks like Miami Beach here. Here's even a tropical, looks like a hurricane or something, heading toward what looks like New Orleans, actually. If I was advising this artist, I'd say put more ice, more water. Make it look less like the Caribbean and more like the Arctic Ocean. Well, actually, the Arctic is losing its ice too. Make it look more like the Antarctic Ocean. Lots of ice. Mars was cold, even was wet. But why doesn't Mars look like that now? The short answer is because it's too small. So here is Earth and Mars shown to scale. And you appreciate that Mars is about 1/10 the mass of the Earth when they're compared this way. It's much smaller. As a result, it has no plate tectonics, less gravity, and no magnetic field. And all of these factors cause Mars to lose its atmosphere and become a cold, dry world. The problem for Mars is not that it's too far from the sun. If Earth was where Mars is, it would still be habitable. The problem for Mars is it's too small. You have to be this big to be a habitable world. This big being at least half the mass of the Earth, we think. Mars doesn't quite make it. And that's why we think it was only habitable for the first 500 million years of its history. Sounds like a long time, but it's short compared to the age of these worlds. So that, in a nutshell, is our understanding of Mars. Started off like Earth, but went through a different evolutionary path fundamentally because of its size. But it's still a very interesting target. So Viking landed on Mars in the early mid-70s to search for life and search for organics. Just as a little historical footnote, Viking landed on Mars the year I entered graduate school. And that had a decided impact on my career. That's what got me interested in Mars and astrobiology and everything. Viking landed on Mars to search for life. What it found was water and the puzzle, no life and no organics in the soil. And so here I am, a first-year graduate student. It's when you think you know everything, when you really know nothing. And I fit that mold. And so I was thinking, well, this shouldn't be hard to figure out. And so I got interested in this whole question of why did Mars have all the elements needed for life, it seemed, but no life and no organics. And I'm still trying to figure that out now. The first puzzle, the one I want to focus on tonight, and the one that's relevant to curiosity, is there should be organics on Mars from meteorites and from any early biology. And the fact that Viking didn't detect any was very peculiar. The solution to that puzzle started really in 2008, many years later, when the Phoenix mission, shown here, landed in the polar regions on Mars. Here's the Phoenix mission. Here's its arm, sterilizing the bioshield. And I added just a note for a slightly different talk. Phoenix landed on Mars carrying 100,000 Earth organisms. We know that. It was calibrated at that. We have sent life to Mars, and many of those organisms are still alive. And we can talk about that later. What Phoenix found was ice in the ground. That was not unexpected. In fact, the site was chosen near the polar regions to find ice. If we had failed to find ice, that would have been an unexpected result. But it found something that was unexpected, several things that were unexpected, but the biggest one, and surprising to us all, was that the salt in the soil on Mars was perchlorate, not chloride. If you go to any environment on Earth, any desert on Earth, and you look at the salt in the soil, the dominant form is sodium chloride, the same salt that's sitting on your kitchen table. In a few rare cases, it's another chloride. Don Juan Pond in Antarctica, it's calcium chloride. Some environments you can find dominated by magnesium chloride. But nowhere on Earth is chlorine not dominated in the chloride form. And we just assumed that this was true on Mars. For 30 years we've assumed it. We finally go and make the measurement, and it's not. The dominant form of chlorine in the soil is perchlorate. Well, what is perchlorate? That's what I asked. I was part of the Phoenix team when it first came out. I'd never heard of it before. Right? Perchlorate, what is that? Now, it's my favorite molecule. And in fact, I'm the self-appointed president of NASA's Perchlorate Club. We have monthly meetings to talk about perchlorate, which is really quite a fascinating molecule. Shown here, it's a chlorine atom attached to 4 oxygen atoms. The chloride would be just the pure chlorine. So here, the chlorine has now got 4 oxygens with it. Well, why is perchlorate so interesting? Well, first, it's used in rocket fuel. The shuttle, you may remember it, used to launch astronauts to the space station. Its solid rocket boosters are ammonium perchlorate. The reason it's used is because perchlorate literally breaks up and explosively breaks up when it's heated to about 350 degrees, that's why it's used in rockets. And that could be, we think now, explain the lack of organics in the Viking results. There was perchlorate in the soil. When Viking heated it up to do the analysis, the perchlorate became highly reactive. Not a good way to treat your samples. Interestingly, perchlorate is also a very strong antifreeze. It's also edible if you happen to be a microorganism. It can be used by many microorganisms as a terminal electron acceptor, roughly equivalent to nitrate. That's very interesting. But it's bad for humans. It interferes with the thyroid, substituting for iodine, and it's toxic. It's a big problem around military bases, in fact, perchlorate contamination. Here's a little example of why it's bad for analytical instruments. I mixed in this spoon a little bit of sugar and perchlorate. This was done for a film crew. And then I started heating it up with a Bunsen burner. When it heats up to a certain temperature, the perchlorate breaks down about 350 degrees, releasing reactive oxygen, which burns the sugar quite spectacularly. This is what we think happened inside the chamber in Viking. As it tried to analyze the organics, it heated up the sample to 500 degrees. The perchlorate became reactive at 350 and literally burned any organics that were in the sample, which is why Viking didn't detect the organics. That's our explanation when we take the Phoenix results and apply them back to Viking. So that's where it was when we landed on Mars with Curiosity earlier, middle of last year. Curiosity landed on Mars in August with an instrument to detect organics. Here is Curiosity being assembled at JPL. And just the same side theme, Curiosity carried 500,000 viable microorganisms from Earth to Mars. These aren't the microorganisms. These are people in the lab. These are normal-sized people, not little models put in to make the vehicle look big. They're not, maybe not as tall as me, but they're normal-sized JPL folks. Shows you how big this is. This is the largest rover by far that we've sent to Mars. It's about the size of a Mini Cooper, so quite enormous. It landed. It was sent to Gale Crater. You see the other landing sites: Viking, Pathfinder, Opportunity, Spirit, and there's Phoenix up there at the edge of the world. Just a little further, and would have fallen off into space. So this is something to keep in mind. We've only had, what now, 7 successful landings on Mars. So we don't know a lot about a place that's as big as all the continents of Earth. We've studied it in detail at 7 landing spots. So here is I can't resist showing this. This is an animation of the landing. And this is an artist animation of what happened on Mars in August when the vehicle landed. It comes in on a parachute and heat shield, just like any other space mission, but as it gets toward the ground, it starts acting in a way that's unique to this mission, where it hovers over the ground and then lowered, it's now past tense, the rover down to the surface. We had never landed this way before. When I first saw this video, I thought it was one of those joke videos, like the beer commercial on Mars, and I started laughing, right? That was not the proper response. This was really what the engineers had in mind. It was the only way they could land something that heavy. And my hat's off to them. It went beautifully. Needless to say, there was a lot of suspense and anxiety over, was this going to work? $2.5 billion down the tubes if it didn't, but it worked perfectly. Really quite an amazing feat of engineering. There was literally not a flaw in the landing sequence as we're watching it beam down 15 minutes after it happened, because of the one-way light time of things coming to Mars. One of the instruments I'm involved in is this one right here. Oops. That telescope that was looking out of the mast, that big eye, is a laser, which I'll show you, the zapping laser, which we can use to analyze rocks. I'll show you the results from that in a minute. This is a photograph of MSL, Curiosity rover, on its way down, on the parachute, being photographed by another spacecraft. So one spacecraft is direct from Earth to landing, another spacecraft is in orbit photographing as it lands on the parachute. Really amazing. In this example, there's no privacy on Mars. Everything you do is being watched. So as I said, the one instrument on the mast is a laser-induced breakdown spectrometer. It's what colloquially we call the laser zapper. We can zap rocks with it. So here's artist's conception of it. It's called the ChemCam instrument, and the PI is Roger Wiens at Los Alamos National Laboratory. The nice thing about this instrument is it's the only science instrument where cameras aren't included, where you can shoot without moving the rover at all, without the rover having to move. You can do analysis up to 7 meters away. We can analyze the rock. So the rover can sit there and zap rock after rock and decide, oh, that's the rock we want to go pick up. And I predict that this has been so useful on this mission that the next rover that we're planning will probably have even more of these remote instruments, because the hardest thing to do with a rover on Mars is move it. It's much easier if you can do the analysis, figure out exactly what rock you want, and then go move the rover. So here's a close-up of it again. There's the telescope that the laser shoots from. It shoots a beam, focuses the beam, and then looks at the light from that breakdown. And this instrument got a lot of press at first because it sounds cool, laser zapper, zap the rocks. And when we started it, there was a lot of blogs and tweets about rocks complaining that they were being shot. And this appeared on the web. Now, this is an exaggeration. It's an exaggeration because the laser light is not green. It's red, it's 1 micron, so you wouldn't even see it. And also the flash is not this big, it's about the size of a pinhead. It's a little tiny pulse. But this is a very fun instrument. It's also very powerful analytically. We have fired this laser over 40,000 times. Zap, zap, zap. We've done over now about 10,000 in separate analysis. Compare that to the other instrument I'm involved in, the GCMS, which has analyzed 2 sites. Done 3 samples at each of 2 sites. And you see why these kind of non-contact instruments are so powerful. And so I'm pushing now for moving beyond just LIBS to things like Raman and fluorescence. The more we can do without moving, the better, which is very different than the way we do field work on Earth. And on Earth, in just about any field environment, the easiest thing you can do is move. It's hard to do anything else besides move. So sometimes I'm in the field, all I do is walk around. Right? On Mars, it's exactly the opposite. The hardest thing to do is move. And so these sort of, being able to do analysis without moving, these sort of instruments are very important, and we're learning that. And ChemCam has shown how useful this can be. And this is an example of the kind of data we get from this instrument. We basically have a glowing plasma, a little tiny one, and the elements glow, and we're seeing emission lines. Essentially, these are like the Fraunhofer lines we see when we look at the spectrum of the sun. We see these emission lines. And we can tell about the elemental composition of the rock. Here is our landing site. I want to show this picture partly as homage to Ray Bradbury, who was a science fiction writer, the author of the Martian Chronicles. If you haven't read it, read it again. I just read it again a few weeks ago. And this looks like a burn mark. This is where the rover landed. It's not really a burn mark. What it is the thrust from the rockets blew away the light, fine-colored material, and that caused the surface to look darker. And so the landing site was named for Bradbury, and it's appropriate in 2 ways, which I don't think we had in mind when we named it for him. One is he was a big fan of rockets. He wrote a book, R is for Rockets, and the first chapter of the Martian Chronicles is called Rocket Summer. He really liked the word rocket and the idea of rockets, so it's appropriate that a rocket landing site should be named for him. And second, he had this thing about names. He thought it was inappropriate to name features that had names of their own. And several of his stories dwell on that. But this is a feature that had no name. It's no feature. This is a big flat plane. The only feature here is the rocket landing site. So it's kind of interesting connection to Bradbury. And if you read Martian Chronicles again, put this image on your screen as you do so. So this is the Bradbury landing site, and here's our tracks as we started off the first couple days. And I like this picture because the Bradbury landing site, and also because here are tracks out to nowhere. It's sort of a call to adventure. Let's go exploring. Drive around on Mars. It's really quite cool. And here's where we've driven so far. We started off at the landing site. We've driven about 500 meters. It's taken us about 200 days. We're ending up here in this bay, and this is the site sample where we've drilled and gotten our most interesting results. A reporter asked me the other day, how long would it take a human to do this kind of field work? Everything we've done. It's taken us 200 days on Mars to cover half a kilometer. I said, well, in a field work on Earth, a human could do this in a couple of hours. Everything we've done on Mars, 200 days, we could do in a couple of hours. That doesn't, I don't mean that to denigrate the value of a rover. The rover doesn't need to come home. It doesn't need oxygen, doesn't need food, doesn't need showers. Well, people don't take showers in the field either. But they do have food and oxygen. But it is, I look forward to the day that humans are on Mars and we can do field work the way we do it on Earth. It'd be very interesting to see how much we get done. But meanwhile, we settle for rovers. Along the way, we saw evidence for fluvial features, water flowing like these. This could be a riverbed, these kind of gravels. This was the first sample we analyzed, this dust pile. It was picked not because it was scientifically interesting, but because it was good-sized sand rather, for cleaning the instrument, the sampling instrument. So we weren't expecting it to be very interesting from a science point of view. This is the scoop full of that sand. And this is the instrument that we fed it to. This is a pyrolysis GCMS. It's part of the other instrument I'm involved in, an organic analyzer. The PI is Paul Mahaffey at NASA Goddard. And notice that it is a pyrolysis unit. Its fundamental mode for analyzing the soil is heating it up. You might think, well, didn't you just tell us the soil is full of perchlorate, which destroys the organics if you heat it up? Why are you sending a pyrolysis to Mars again? That's a very good question. And the answer is, we learned about the perchlorate on Mars in 2008. This instrument was proposed to NASA in 2004 and built in 2006. It was supposed to fly in 2009. It was delayed to 2011. The timing just didn't allow us to actually know the results of the Phoenix mission before this instrument was designed. It's one of the problems of the space business. It takes so long to get out to these worlds and to build these complicated missions that you have to lock in designs sometimes 6, 7 years before the mission. And so anything you learn after that, it's too late. Fortunately, by chance, really, this instrument has 3 modes which should allow us to detect the organics even with the presence of perchlorate. I won't go into detail, but basically we can scan in temperature, which Viking couldn't do. We have 2 liquid samples, 2 types of liquids that we can use to extract the samples. And we also have a combustion mode. So it's a more powerful instrument, so we will see organics where Viking did not. And we will be able to see through what I call the perchlorate wall. So what will we see? I think what we'll see, first and foremost, is organics that have come from space, from meteorites. We were lucky in California last year to have 2 meteorites come across the sky. One landed near Sacramento, and it was full of organic material, like the Murchison or Tagish Lake or Orgel meteorite. It was organic rich. Those kind of meteorites are falling from space all the time on Earth, on Mars as well. They should be a source of organics. Meteorites fall in, they come apart, they're processed by UV, they produce CO2 and methane, and they should leave some level of organics in the soil. We should see organics in the soil, if from nothing else, from meteorites. And that's a pretty interesting prediction. We know what these organics are like. They're mostly, for example, things like PAHs, polycyclic aromatic hydrocarbons, but there's also amino acids in meteorites. So we should see those in the soil. Failing to see them in the soil would be quite a puzzle, especially if we can get past the results of perchlorate. So here's the results from the first analysis. We saw that there was a lot of water in the soil. We saw CO2, probably from carbonate and any organic material, being burned by the perchlorate. Here, this oxygen pulse is the perchlorate decomposing, just as expected. This is the monster in the soil. This is what's defeating our attempts to measure organics, is this release of oxygen. This isn't just O2, this is O, reactive states of oxygen and chlorine that are attacking the organics. And we have to get around that perchlorate wall, that perchlorate barrier. Now, Viking could only sample at 500 degrees. It heated up the soil to 500 and then sampled, the damage is done. They didn't see anything. But we can scan in temperature, so there's a possibility that we will see organics before the perchlorate decomposes or after, at lower temperature and higher temperature. I'm particularly keen on this higher temperature organics because we have some contamination in our instrument which comes out at low temperature. But here, it's pretty clean. We're past the perchlorate. That's where we should see organics, and we know that meteorites have high temperature organics in them. So that's what we should see. We haven't seen it yet, but this is our first sample. We're now doing our second sample at this site. And I love this picture because it looks so familiar. I've been to this place, driving from Los Angeles to Las Vegas. It's a wonderful drive. You pass many, many scenes that look just like this in the Mojave Desert. It looks so familiar. It looks so Earth-like. It's part of the subconscious allure of Mars, at least for me, is looking at these images and I think, I could be standing there trying to see if I get cell reception. And so here is looking down on that sample, on that surface. We saw these cracks. These cracks were full of gypsum, indicating that this was probably mudstone deposited in water. Very interesting, probably the most interesting rocks we've ever stood on Mars. We drilled into it, and it was confirmed that these were interesting rocks. So here is before and after drilling. Before and after drilling. And the amazing thing is that the ground is red, the stuff that we drilled up is gray. And you think, is that the level of sophistication of your analysis? Yes, at this point it is. Red and gray, red and gray. Everywhere else we've ever analyzed on Mars is red. Viking was red, Phoenix was red, Spirit and Opportunity were red, with some minor exceptions of some of the stuff in one of the tracks. It wasn't very interesting from a biology point of view. But we always thought that below the red would be gray. And we thought it for two reasons. One, from first principles. The gray is a layer of rust. Imagine a rusty nail. You cut the nail and the inside is still gray. The gray is a surface phenomenon. And second, the meteorites from Mars are all gray. There's a meteorite in the building, in the lobby of my building. For years, I'd walk by it and something was bothering me. This meteorite from Mars, and yet it's gray, and yet I know Mars is red. Why is that? It's because the meteorites are coming from craters that are deeper down. So we knew that Mars was gray. We just never seen the gray stuff before. Well, here we drilled our first hole on Mars that's ever been drilled, only about 10 centimeters, and we hit the gray Mars. Now, I was very excited when I saw this. Gray Mars, finally. We knew that the sample we'd analyze was red Mars from the sand dunes, and now we were going to analyze gray Mars. So I went on, I was being interviewed by a TV or a documentary producer who's doing a show, and I firmly predicted that we'd find organics in this stuff. This is the gray Mars. This is what we've been looking for. If this was Earth, and we'd found a mudstone like this and drilled into it, and we went from oxidized to clearly reducing material, we would be sure that it would be loaded with organics. So we've done this analysis. We're doing this analysis right now. Unfortunately, Mars moved behind the sun, and we're losing link with it for 3 weeks. But when it comes back, we'll complete the analysis. We don't have the answer yet. When Charlie first asked me to give this talk, I was sure that by the time I came here, we would know the answer to this question. Are there organics in these reducing samples on Mars? All we know is that this is clay-rich and that it's reducing. But the organics, we haven't been able to tease them out because of the complications of the perchlorate and other complications that make it hard for us to pull out the Martian organic signal. The perchlorate and the contamination from one of the liquid reagents that we brought with us is what's making it hard for us to find organics that are Martian, and with the confidence that we're willing to say, hey folks, we found Martian organics, because that will create quite a scientific interest as well as media frenzy, because people automatically assume organics means life. Here is the sample up close getting ready to fed. But organics do not necessarily mean life. So I want to quickly talk about that for a second. We will find organics on Mars, but that is not the same as finding evidence of life. We know that life is made of organics. That's true. But we also know that there's non-biological sources of organics from meteorites and from examples, Miller-Urey type experiments in the lab. And the question is, can we tell the difference? But and I'll make a prediction, when we announce that organics are on Mars, there will be this immediate equation with organics and life. And this is not just in the public. Even the scientists who aren't in this particular field, but the geologists and astronomers also have this confusion between what is organic and what is biological. And it's my personal quest to sort this out. You go to the grocery store, and they have the organic section. I try to explain to them that that's mislabeled, that everything in the store is organic. Not only is it organic, it's biological. Except the salt, where they have organic salt. I say salt cannot be organic. It's a mineral crystal, sodium chloride. The clerks don't always appreciate my helpful advice. But it shows the confusion of this word organic. And when we find organics on Mars and announce it, there's going to be the same confusion. The word is used in many different ways, and even in astrobiology, it's thrown around as if it's synonymous with biology. So we'll see. But how do we tell the difference? This is the next step. The first step on Mars is finding organics. The next step is how do we tell the difference between these organics and these organics? How do they differ? Is there some signature we can use to tell the difference between organics that come from a biological process and organics that come from a chemical process? Interestingly, there is. And the best example of that is amino acids. Biology uses only left-handed amino acids to produce proteins. It does not use right-handed amino acids. So if you have a sample that's biological, all the amino acids in it are left-handed, if they're from proteins. Whereas a chemical synthesis produces roughly equal amounts of both. So if we were to go to Mars and find a sample that contained only one type of amino acids, be it either left or right, it would be indicative of a biological selectivity. It's kind of like driving on the road. You got to drive on one side or the other. Here you guys drive on the left, perfectly okay, as long as I remember to look all three ways before I cross the street. You know, elsewhere we drive on the right, perfectly okay. You don't want to do what they do in Italy where they drive on both sides of the road because this is inefficient. So biology chooses one side or the other. Chemistry doesn't. And this is one example, and there's many, of how biology is selective in what kind of molecules it uses, and chemistry is not. There's other examples in isotopes and sugars and so on. That's what we might use to detect the signature of life, even if none of the life forms are still awake, still alive. So a specific proposal is to search for amino acids, look for the ones that make up Earth life, hope that some other biology might have a different set, and even stranger biology has a set that no overlap with Earth. And chirality is part of this phase space here that we're looking at. So that's what we might hope to find on Mars with the sample. I want to now briefly turn to my other favorite world to search for life, which is Enceladus, a little moon of Saturn. What became a star in astrobiology when it was discovered by Cassini in 2005 that there's jets of water coming out of the south polar region of Enceladus. The flux of this water is equal to the flux of Old Faithful geyser. So a lot of water issuing out constantly. It's estimated that if this flux continued over the age of the solar system, Enceladus would lose 10% of its mass. So it's not going to deplete Enceladus quickly, but it's a lot of water. And the Cassini analysis indicates that this water is coming from a liquid reservoir of slightly salty composition. And this was determined by a close flyby where the spacecraft flew so close to the plumes it could literally catch the little droplets coming out. So we think that it's a pressurized liquid water pocket venting to the surface, a geyser model. This plume contains ammonia, biologically available source of nitrogen, and all sorts of organics. So we have liquid water, we have organics, we have biologically available nitrogen, and there's obviously a geothermal energy source driving the plume. Check, check, check. All the big requirements for habitability are there. So we're pushing right now very hard a mission that would fly through the plume of Enceladus, grab a sample, and bring it back home to Earth for analysis, using the same approach as Stardust, which flew through the plume of a comet. We would fly through the plume of Enceladus. So that mission, something we're studying now. Enceladus is very far away. It's a mission that would take 25 years before the sample comes back. So for those of you that are in grade school, look ahead to working on this as your thesis project. I want to turn to the question, what if we find alien life? Last few slides dwell on this. This came up, interestingly, I got a phone call from some students at Michigan. They said, can we ask you a few questions, interview? I said, sure. The first question is, are you optimistic we'll find life on other worlds? And I said, well, obviously I'm optimistic because that's what I'm doing for my job all the time. My optimism is not a good indication of its probability. Then the second question was, are we ready to find alien life? Are we ready? I realized, no, we're not ready. It's like the dog chasing the car. Is the dog ready when they catch the car? Do they know what they're going to do with the car after they catch it? Right? They don't, right? We're in that same position. We're searching for a second Genesis. Well, what if we find a second Genesis? What does this mean? Does it change anything? Should we be doing anything differently? So I started thinking about it. Yeah, it does matter. If there is a second Genesis, then that may change our approach to life and our approach to exploration. And so that, in summary, what I think is if we found another type of life and it was a second Genesis, we would now have three different sets of moral considerability. And what I mean by that is there are two sets right now, roughly speaking, humans and life. We all realize, and it's encoded in law, that humans are special. We also have an intuitive feeling that life is special. Life is more special than non-life. It's not, unlike humans, it's not unitary, where all humans have the same specialness. It has a sliding scale. We all like polar bears more than we like microorganisms. except maybe Charlie. And we kill microorganisms all the time. We brush our teeth and wash our hands, and that kills millions of microorganisms, and I encourage you to continue doing these things. Right? But we all have an appreciation that life forms a set of special entities. Well, now we would have another set, life too, distinctly different. The conclusion would be that microscopic organisms, which score low on moral status based on the usual criteria, would have high moral status if they were on Mars, if they were the sole representatives of a completely different set of life, different type of life. I'm not going to develop this idea in detail. It's something that actually Charlie and I have talked about at length and is interesting to consider. But I want to say that it has implications for what we do right now. It's not just a purely academic exercise. And I argue that the implications right now is that we should make sure that everything we do on Mars is biologically reversible. That is, we should not irreversibly contaminate Mars. That's why in the slides I had keeping track of how many organisms went on each mission. We have their names, we have their numbers. So if we want to, we could go back and collect them all and bring them all back to Earth. They aren't spreading in the environment because the environment is lethal. We need to be able to do Control Z. Control Z is my favorite metaphor from the computer domain. Undo it. My computer, every time I'm about to do something dumb, it pops up a box that says, do you really want to do this dumb thing? You can't undo it. Right? And I think that's wonderful. I wish my life had this feature. You know? Do you really want to do this? You can't undo this. But so far, everything we've done on Mars, we are able to hit the Control Z button and undo it. We have not done any irreversible contamination on Mars, and that ought to be an explicit policy. Why? To preserve the options in case we do discover a second Genesis. So the questions of, does it matter that we discover second Genesis? The answer is yes, it does matter. And it matters how we make the exploration now, if we think it through. So it's been an interesting exercise to follow through. I now want to end with a final thing that we're doing that's got nothing to do with the rest of the talk. And you may think it's in violation with this notion of preserving life for second Genesis. So we're exploring if Earth life could grow on Mars or the Moon. We're building a plant growth module right now that we're going to fly on the Moon on one of the Google X Prize missions. We'll be the first organism to grow, live, and die on another world. The scientific purpose will be to test germination in lunar gravity and radiation. And then ultimately, we want to fly it to Mars. But we would fly it to Mars and grow it on Mars in a way that's biologically reversible. So while at the same time we're determining if Earth life could grow on Mars and colonize Mars, we're holding open the option that if we find Martian life and Martian Genesis, we can undo the Earth effects and let that Martian life proceed. So our motto is, fly me to the moon. And then this is my last slide, and it's my clue to ask for questions, and thank you for your attention.
Thanks, Chris. Fantastic lecture, entertaining and educational as usual. We have time for about 10 minutes worth of questions if anyone has questions. Yeah. Yes.
Wait for the mic because they're recording everything. And
I can bellow.
okay, go.
If you're sending soil and plants to Mars, I don't understand how you can then do the Control Z afterwards because you deliberately planted life there. So it might not be replicating and growing, but it could go into dormant states and survive.
Yeah. What we'd like to do is do greenhouses on Mars. Because the Martian environment, though, is inhospitable, they have to be enclosed. So everything that's growing would be inside the bubble. Anything released would be exposed to the incredibly biocidal UV radiation on Mars and be killed. So basically,
Crash on landing.
pardon?
And if you crash on landing.
If you crash on landing, again, anything that, and several missions have crashed, everything that's released into the environment is killed. Only those things that are shielded from the UV remain viable. Dormant, but viable. And so at crash sites, you'd have to find the pieces and scoop up the dirt around it as well. So it's not trivial to do the decontamination, but it's not exponentially hard. The way to understand it, I use an analogy with rabbits. Say you have 2 rabbits and you take them to a continent that starts with A, Australia. You let them go. And you take 2 other rabbits to another continent that starts with A, Antarctica, and let them go. And then many years later you decide that you want to reverse that. In Australia, it's exponentially hard to do so, as the Australians are finding out. In Antarctica, you'll just find 2 dead rabbits not far from where you left them, right? It won't be trivial to find them, right? But it won't be exponentially hard. It'll be linearly hard. And so the point is that this contamination is contained within the capsule, and the crash site is contained within the crash site. And so the problem of decontaminating is still linear. If Mars was habitable, or if you were to drill down to an aquifer which was habitable, then contamination would be exponentially hard to, and that's what I would call non-reversible contamination. So there's a lot of, as you drill deeper into the question, there's a lot of subtleties that we need to think through. But the fundamental feature of Mars that allows us to think this way is the biocidal bath of UV that coats the entire surface, wavelengths down to 190 nanometers. Question here.
If you found a second Genesis and you took it back to Earth, how would you keep it?
Right. This is a really good question. The question was, if you found the second Genesis and you brought it back to Earth, how would you keep it, control it? This is an important question because we'd have to worry that if we brought alien life to Earth, it could be, 1, infectious. That's unlikely. It could be hungry for the things that we're made of, like locusts. That's unlikely, but not impossible. Or it could be friendly and just grow everywhere, like kudzu or zebra mussels, right? Or rabbits in Australia, where the problem is an ecological one. That's entirely likely. If I lived on Mars and had a chance to come to Earth, I would jump at it. Right? So in fact, NASA's policy, not just set by NASA, but set by the Centers for Disease Control and the Department of Agriculture, is that anything we bring back from Mars has to be strictly quarantined until we determine that there's nothing alive in it. Because we would not want to release something into the environment that we didn't know for sure was harmless. In fact, I don't think we'd ever want to release it in the environment, period. So we would always try to separate Mars life from Earth life. Some people have said that, in fact, the way to study it is to go to Mars and study it there, not to bring it back to Earth. So this is an important question, and we don't really have the answer to it yet. And part of the reason we don't have the answer to it yet is because we're not serious about doing a sample return. Once we get serious about doing a sample return, we're going to have to get serious about solving that problem. The way that we're planning on sample return now, it might be the time you're in grad school, so keep thinking about that question. Question here?
Yeah, our definition of inhospitable has been changing over the years. What we thought was impossible 50 years ago is very much common sense and common practice now in PCR, for example. I'm just asking whether we have properly assessed what's inhospitable in Mars. And secondly, going back to what the young lady here is saying, back to Darwin himself, if we send something there, we are really good life, we're very capable life. If we go there, can our things kill the things already there? Is that a possibility? If there is something down in the aquifers, for example. I know that we're not going anywhere near them, right. But is that a remote possibility that we could kill things that are already there and we wouldn't find them when we try to find them?
Yeah, this is a good question. There's 2 parts to it when applied to Mars. One is, how do we understand habitability? As I was indicating, what I think is the barrier to Earth life surviving on Mars is the UV radiation and the lack of water. Those 2 factors, in that order. UV radiation is more serious, lack of water secondary. If there were organisms on Earth that could survive those conditions, it would change my logic entirely. So if new information came in, then I would change my point of view on that. But right now, our understanding of life is that there's no organism on Earth that could survive in the Martian environment as it is, in terms of the UV radiation and lack of water. But that's something we have to constantly update our thinking. That's the great thing about science. You get to change your mind and contradict yourself, and it's a feature, not a problem. The other point was, could we contaminate Mars in a way that is detrimental to organisms that are there? And yes, that is a serious concern, and it's part of this, why I keep track of these microorganisms and where they are and how many are in each spacecraft. And as long as we're on the surface in this UV bath, I think it's okay. But when we start drilling, I agree with you. We got to worry about that and drilling in aquifers. And then we have to get serious about sterilization. When NASA sterilized the Viking missions, it's sterilization in quotes in my mind. It's called, technically the euphemism is dry heat microbial reduction. But if I was going to a hospital and they had a sign, we sterilize using the NASA method, I would head as far away as fast as I could, because that is not sterilization, really. It's not the standards of autoclave. Right? So if we're going to drill, we're going to have to do medical standards, not NASA standards for sterilization. So the biological reversal principle sort of cuts both ways. It makes it easier if you're working on the surface. Don't bother. The environment does it for you. But if you're going to the subsurface, we need to upgrade our sterilization standards from the Viking level, dry heat microbial reduction, to medical quality sterilization, where it's really sterilized, which means autoclaves and things like that.
Just picking up on that, how much can you learn from, say, doing things on like Lake Vostok and so on, where contamination is exactly that kind of problem?
Yeah, it's a good question. The question was, how much can we learn from Lake Vostok? Lake Vostok is a lake in the bottom, underneath about 4 kilometers of ice in the polar plateau. And the Russians have drilled into that lake for the first time last year, and this year they retrieved samples. So these are environments that may be isolated from the rest of the biosphere for millions of years, maybe, maybe not. And so we would like to sample them without contaminating them. And there's a lot of examples of this in the Antarctic. We did it in a lake called Lake Vita, which was only under 19 meters of ice, but it was sealed and isolated. And here we are trying to do exactly what I was saying, rigorous sterilization of everything that goes down the drill hole. In Lake Vita, as the drill hole went down, it went through a UV bath. We used only distilled clean water. Everything that went down the hole was bathed in biocidal UV radiation before it went down the hole. So we're learning how to do these here on Earth, and maybe that knowledge will apply on Mars. Even more importantly, on Enceladus, where we know that there's a habitable environment. There we know that Earth things could grow. We could contaminate that, and it could be swarming with bugs easily. So we have to up our standards for sterilization when we start approaching environments that have water and are habitable. It goes back to the statement, we're searching for habitability. Once we find it, we're not ready to explore it because we don't have the technology to send spacecraft clean enough right now. We need to develop, we don't know how to autoclave a spacecraft. They won't work after you take them out of the autoclave. So we've got to figure that out.
There was a proposal in the media a couple of months ago to send a married couple on a loop around Mars. If you had the resources to do that, what mission would you choose to do rather than send them nearly to Mars and then not have them land and bring them back again?
Yeah, well, that's a good question. There's two parts. Let me answer in 2 ways. One is, I know that the group that is doing this, and I emailed them when I heard it, and I said, if you go fly by Mars, we have some instruments we'd like you to take along and get some data. You know, something to do on those long Martian nights. Because after 400 days in space, you'll be tired of each other. But seriously, or maybe not so seriously, I once was asked by a reporter, if you could go anywhere, if you had your own little space-mobile, Spiff the spaceman, and you could fly anywhere, where would you fly? My answer, without a hesitation, was through the Plume of Enceladus. Now that's much harder than sending a couple to Mars, but that's where I'd really like to go. If I could fly and had a little spaceship, and it could go fast, I don't want to spend 500 days on this trip. Where I would want to go is the Plume of Enceladus. I think from an astrobiology point of view, that's the most interesting target in the solar system right now. But no one's thinking about that in terms of sending humans.
My question might betray my ignorance because I'm not a scientist, but I wanted to ask about your proposed planets and worlds for where you think there might be life. If this is an organism with entirely different biochemistry to ours, how do you know where they'd be able to survive or where would be inhospitable to them?
Yeah, that's a very good question, and it's a question that scientists should be asking too. But it's a hard question because, as I told you in the very beginning, we only have one example of life. So it's very difficult to make really sweeping generalizations about one example of life. In fact, William Baines, among others, have looked at the possibility that life could be based on completely different chemistries. Right? But it's very hard to get NASA to fork out $500 million to search for a completely different type of chemistry. When you get up in front of the peer review and they say, well, what are you going to look for? You go, well, I don't know. We'll know it when we get to it. What measurements are you going to make? Well, we're not sure, but we might try this and this. They go, next, and they give the money to geologists who want to study volcanoes. Right? So it's, from a practical point of view, it's very hard to know how to search for a different type of life. The one exception, I think, is Titan. Because here we have a story that's different, but not too different. There's organics in the atmosphere, so carbon is available, lots and lots of carbon. There's a liquid, there's energy, there's nitrogen, there's a liquid. The difference is the liquid's not water. So it's like one step away. It's a big step, but it's one step away from what we know. So we can take that. But it's a kind of a catch-22. We have only one example of life, so that's what we look for, but that doesn't mean there can't be other examples of life, but we don't know how to look for it. So my approach, sort of the practical one, is, well, let's search for what we know how to look for. Let's keep Titan in mind, and let's see what happens. And as we learn more about life, maybe we will develop a broader understanding of it as a phenomenon, as a diverse phenomenon, and then we'll be better positioned to study different types of life. You know, it's a question I hope that will be resolved by new data. Finding another example of life will help us understand what to search for from then on.
One more question.
One more question right here.
What's our success rate for landing devices on Mars? How many have crashed and how many have made it there in one piece?
Yeah, that's a fun question. I used to say, the question was how many spacecraft have crashed on Mars and how many have landed? And when I used to give talks, I used to say, Mars is so popular, we crash more spacecraft on Mars than we send to all the other planets put together. But I decided that was bad humor for a NASA employee to be saying that. So now I don't mention the crashes, since some of them are NASA crashes. Roughly speaking, about half the spacecraft that the world, Earth, has sent to Mars have failed. Half. Now you think, well, that's pretty bad. Can't you guys do better than that? But in our defense, I'll point out that a baseball batter who hits the ball 50% of the time, is batting .500, would be in the Hall of Fame, right? And if you're batting better than .500, it means you're in the wrong league. So the fact that we're batting .500 means that we're doing something that's very hard to do, and so batting .500 is good. It's kind of a apology for failure, but I'll stick with it.
I'd like to thank Chris again for a really spectacular talk and interesting answers as well, and to thank him for coming all the way to Edinburgh. Unfortunately, we couldn't invite you to Enceladus, but it's cold here, so at least it's a first step to Enceladus.
Yeah, nice and windy.
And windy. I'd like to just finish off by handing over to Claire, who will say thank you from the museum and finish off the evening. Thank you.
Thank you. I just wanted to say, on behalf of the museum, first of all, thank you to the University of Edinburgh for giving us this amazing opportunity to piggyback on your event this afternoon and to open it up to some members of the public to have the chance to hear Chris speak. A truly fascinating, mind-blowing almost lecture this evening, one of the days that it makes me very glad to be doing my job here. We had a lecture last Thursday evening, which I think some of you were at as well, where we were hearing about the first footsteps, the first footprints, Australopithecine footprints found on the Earth, and then to see the tracks of the Mars rover, the Curiosity rover on Mars. Very strange connections, but equally fascinating. Without further ado, thanks once again to the university and to Chris McKay. Thank you very much.
This production is brought to you by the University of Edinburgh.